Hey everybody! I thought I'd whip out a quick video to try to wrap up this movement section so that we can move on to the defense section this coming up week. So I'm going to try and make a quick video and then we'll do some assessment in class. I'm also trying to take into account all the things we talked about on Thursday and see if I can make some changes to the class to make it more effective for everyone. So hang on for the ride while I try to do this. Okay. So in this last section of movement, we're talking about others. So we've talked a little bit about animals. We've talked about some generalities. Now let's talk about a little bit about plants at the end, but we'll start out talking about bacteria and fungi and some other things. So in this first learning objective, it's describe the structure and function of the bacterial flagellum. So some things you should maybe review from a previous class is that when it comes to the bacterial flagellum, okay, this is a structure that's composed of a number of different proteins, and there's a schematic here on the right-hand side that you're seeing right here. So this is, again, a stylized diagram, and despite this sort of curvy structure here, it's actually a fairly rigid structure that will help to affect movement. And you're going to see both in class later on Tuesday and a little bit here that there's a variety of different structures that are very similar for propulsive movement. So there's flagella, there's cilia, and even flagella can take some slightly different conformations to them. Anyway, in the bacterial flagellum that you're seeing here, it's a rigid structure composed of several different proteins. It is powered by a molecular motor. So interestingly enough, this is not driven by ATP. It doesn't expend ATP to move. It's a proton powered. And you'll see this in a little bit more detail in a video I'm about to show you. But you can see the complicated structure at the base of this thing that's actually going to make it turn. So it doesn't just whip around as many people think. It actually twists. And it can go both in the clockwise direction and it can go in the counterclockwise direction. Although when it does that, the type of movement that then is expressed is not the same. So it's not just going forward or just going backward, and it can whip its flagellum in either direction. So let me show you what I mean. So here's a movie. There's no video or there's no audio to this movie. So this is showing you a little bit of part of that movement. So what you'll see is that it's just going to do a close-up here of the base of the motor. It's proton powered. And as that motor fires up, you can see it's now moving the flagellum. It's kind of got some S-curves in it, but it's moving it in a clockwise fashion. But literally on a dime, it can automatically switch, and then it'll start turning in the counterclockwise direction. Now, what those different movements, clockwise versus counterclockwise, they correspond with the mode of movement that these bacteria will display. Oh, poop. Hang on. There we go. So, if the motor is turning in a clockwise fashion, you get what's called the running mode. And so basically, it's going in a roughly straight line direction. When it turns around and it goes that's counterclockwise. When the motor is turning in clockwise, it does what's called a tumbling mode. And so what's happening is when bacteria are searching for a nutrient resource, right? So it's going to be out there somewhere. They affect what's called a random walk style of locomotion in trying to find that nutrient source. And so they'll start going off, the motor will be going counterclockwise, they'll move in a straight line direction, and then at periodic intervals, they'll then switch to the clockwise rotation, which then puts them in this tumbling mode. So they're alternating between straight line, and then tumble, and then straight line, and then tumble. And what that does is the tumbling mode is what causes them to basically change direction. And that's what these diagrams at the bottom are supposed to be showing you. So every time it changes direction, so it goes forward, tumble, changes direction, forward, tumble, change direction, forward, tumble. So and then by virtue of alternating between running mode and tumbling mode, it's this random walk movement, which is supposed to help them be able to find that nutrient resource. And then once they do, they'll begin to change their mode of movement so that they zoom in on that nutrient resource. So it's a pretty effective thing for a simple organism to engage in. Okay, the second objective, so now let's move on to some other organisms. So first we're bacteria. Now we're going to talk about some unicellular eukaryotes. So previously referred to generically as protists, but we now know that that is not a monophyletic group. So we're going to briefly talk about amoeboids, because it turns out there's several different organisms that exhibit amoeboid locomotion, but they're not all amoeba. We'll talk about ciliates, sporozoans, and flagellates. And in class, we'll talk about the difference between cilia and flagella. Okay, so the amoeboid movement, we've already talked about a little bit earlier in the semester, but just to remind you, so this requires ATP, so it is energetically expensive. They're highly characterized, and you can see the video to the right too. It's going to go much slower. So it sticks out these finger-like projections called their pseudopods, and then they move, and we're not going to go into the gory details of how that movement is affected, but you can summarize it by just saying that they do have actin and myosin filaments in their cytoskeleton, and the interactions between those basically pushes the plasma membrane, the edge of their pseudopodia, in some particular direction, and then it'll form these finger-like pseudopodia projections, right? And then I say here that the cytoplasm streams along behind. There are videos that you can see in Khan Academy that shows you the gory details on how they move. What we want you to know is simply that various kinds of protist-like organisms, they can move, they move in different ways. And we're going to give you sort of the highlights of how they can move. So one of those types of movement is amoeboid here. And I believe I showed you in a previous lecture an amoeba that ate I think it was some sort of single-celled algae, and I made a joke about the algae screaming. Okay, now you got pseudopodia. You also have the ciliates. So in this case, what you should be able to see in this video here, but also on this cartoon, is that ciliates are characterized by, their bodies being covered in cilia. In this case, you're actually looking at a particular kind where it has like pairs or triplets of groups of cilia, but there's all different forms of ciliate organisms. This type of movement does require ATP, and I'm going to explain the difference again between cilia and flagella and their movements in class. It's hard to do here in front of the screen right here. But it is energetically expensive. These cilia are composed of microtubules, which are composed of the protein tubulin that move the cilia to create movement. And the great thing about cilia is they can move you in just about any direction, forward, backward, sideways, and I'll show you how in class. And then, of course, you have flagellae. So also energetically expensive. So all three forms, so cilia, flagella, and pseudopodia, uses ATP to make movement happen. Similar to cilia, not surprisingly, composed of microtubules, which are composed of tubulin to create that movement. But there is a big difference between how flagella move and affect movement and how cilia move and affect movement. Or locomotion, I guess I should say. By the way, don't be fooled. Like the numbers of flagella that you might have can vary, and the size of the flagella can vary as well. Okay, so now let's move on to the third objective. So we're going to briefly describe mechanisms of dispersal in fungi. Hey, and guess what? Newsflash, fungi don't really move. In fact, they don't move at all. So the only time when you would really see movement in most fungi is when they're reproducing, and that's by broadcasting their spores. Fungi can do this in a variety of different ways, and this video is showing you one, which is, it's a relative of puffballs. This is a different form of fungi, but you can see puffballs here all around Michigan certain times of the year. And so if you touch them, or in this case, if you have a really heavy drop of water that hits, it'll cause disruption to the insides of the spore-bearing capsule, and you can see it. It's basically woofing out spores, which will then be broadcast on the wind, or if there's other organisms around, they'll carry them off as well. There's another video I'd like to show you too. In this case, this is a different form of broadcasting, so they can literally shoot them out almost like arrows. and at really high velocity to get their spores dispersed across the environment. So this video is gonna, I don't have any audio to go with this video, but they're basically trying to show you how fast it is and that it's faster than a bullet. Oh. So I guess that means fungi are more badass than guns. There you go. All right, I'm going to move on to this next part. Nope, there we go. Okay, we're going to talk a little bit more about fungi in class, but let's move on to learning objective 4. So now we're going to talk about tropism in plants. Now check this out. So here's a light, and watch what these little seedlings are going to do. Now, of course, this is sped up in time, but notice that they clearly do move towards the light, especially when the ambient other lights are turned off. It's rather mesmerizing, isn't it? You can just sort of, tune out while watching this. Oh, and then once the light's gone overhead, not surprisingly, they're going to move up. So they're not going to move just towards that one single source, but they're going to move up. By the way, notice they grew a little bit too. So notice how at the tips of these things, they're all about the same, but there's just a little bit of growth happening at each tip that gets bigger and bigger. Dun, 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 dun. I don't know what the complete period of time this thing is under light. But it's clearly making new growth at each tip. Some of it's pretty considerable. Look at that. And then you can see the new growth at the tip of each of these shoots. So pretty impressive. Okay, so what's going on here? So this is called the phototropic response. That's a response you're already familiar with. But just to review these definitions. So tropism is growth or turning movement of a biological organism, in this case usually a plant, although you can see it in fungi too, in response to an environmental stimulus. And then of course phototropism is the directed movement in response to light, as opposed to gravitropism, which is the response to gravity. Plants sense and respond to a specific narrow range of wavelengths in the visible spectrum when they engage in phototropism. So we're going to talk more about this in class, but what I say here, and I'm showing you this diagram at the bottom that shows you not the actual wavelengths, but it's showing you the different color bands, if you will, that you find in the light spectrum. So plants sense and respond to a specific narrow range of wavelengths in the visible spectrum. Charles Darwin is famous, he and his son are famous for conducting experiments with plants, showing that in fact, so they were some of the first ones to show that plants will move in response to light, and generally, primarily to blue wavelengths. So if you shine different wavelengths at them with a particular kind of light and a filter, they'll only move towards the blue light. And actually you can see that here. So later, only fairly recently in recent experiments have we figured out how they do this. So they have these genes that are referred to as phototropins. And those are the genes that make plants able to detect and respond to blue light. And so what you're seeing in these pictures, you can't see the light. So they've put a blue light source over to the right-hand side. And what you can see here is the wild type. This is Arabidopsis, this kind of classic plant model system that's used in all kinds of experiments. And so what's happening here is the plants are actually moving, bending towards the blue light source. But in these FOT1 mutant Arabidopsis lines, they're not responding to that at all. They're just going straight up and down. So photoreceptors is what allows them to detect light and respond. Okay, so what's the response to blue light? So what are plants actually doing? So this is a signal transduction cascade that gets triggered. And so I'll show you some videos of this, but basically chloroplasts those little green cells, right? They're going to move within leaf cells to positions that optimize light absorption, right? So if if this is a cell, right? wherever that light source is, if it's like out that way, then all the chloroplasts are going to gravitate or move towards that part of the cell so that they can maximize their ability to detect and utilize that light. What also will happen is stomata will open to allow carbon dioxide to diffuse into cells as blue light triggers photosynthesis. Remember, this is energetically expensive, so stomata are going to open to facilitate photosynthesis to occur. And you will also get stem elongation, okay, growing towards that light. But we need to talk about how that happens. So how do cells elongate? So previously we talked about several different plant hormones, and auxin is the one that's the most important in this particular instance. So auxin is produced actually both in shoots and roots, but in this case we're talking about shoots. So it's produced right at the tip of a young shoot, which is called the coleoptile or coleoptile, I've heard some people say, so right here. So what happens is that gets produced and then it gets distributed throughout the plant shoot, that young shoot, right? But it's asymmetrically distributed. So at first, it's basically distributed throughout the plant, but on the sunny side of the plant, this side, that auxin is going to get broken down. So as the sun moves, that shady part is going to get warm, the auxin is going to get broken down, and then where the auxin remains, so in this case on the left-hand side of the shoot, it's going to cause the cells to elongate. And by virtue of causing these cells to elongate and these cells not being affected at all and not changing shape, that makes the shoot bend towards the sun. Pretty amazing, right? So auxin promotes cell elongation on the shaded side, leads to the bending of the shoot towards the source. I just said that. Okay. We're going to drill down and get into this in a little bit more detail. Okay, so hold on for the explanation here. So here's what's actually happened if you get down to the micro level a little bit. So auxin is going to bind to proteins found in the stem and in the leaf on the shaded side, right? So the signaling pathway that's triggered increases the number of proton pumps in the plasma membrane. So here you have all these protons getting pumped out of the plasma membrane out to here, okay? And then, here we go. So protons pumps use energy to pump protons out of the cells, and it's going to acidify the outer part of this cell wall, okay? So that lower pH and the activity of expansion, so expansins are going to come into the area, and it's going to loosen, so this acidification causes loosening of the cell wall structure, okay? Now, because the interior of the cell has become more negative, because remember those protons got pumped out, that's going to lead to potassium ions moving into the cell. So that's what you see going on here. And as the concentration of solutes inside the cell, oh, where'd my cursor go? As the concentration of solutes inside the cell, see that happening here, increases, water is going to diffuse in through the plasma membrane, and it's going to cause turgor pressure, and it's that turgor pressure that causes the cells to elongate, okay? So you got protons pumping out, acidifies the cell wall. That acidification and the interaction with expansion is going to cause the cell wall to loosen up, to get less rigid. And then with the changing solute concentration inside the cell and water being pumped in as a result of that, it's going to cause turgor pressure, which then makes the cell expand, and that's where the elongation is going to come from. Okay. So now let's talk about a different trophic response. Let's talk about gravitropism. So we already said this is the ability to move or grow in response to gravity. So of course roots respond to gravity by growing down. Shoots actually do respond to gravity as well, and they can grow up. So if we're focusing just on roots for a moment, so the end of roots tips have this protective root cap, and that's what it's trying to show you right here. Here's the root cap. And there's these gravity sensing cells that contain starch granules, that's what you're seeing kind of highlighted here, located at the center of the root cap. So these are your gravity sensing cells. So if we kind of zoom in on a schematic of what that would look like, there are these things called amyloplasts. So these are dense starch storing organelles. Okay, these are here, these white cells here. And these guys respond to gravity by being pulled to the bottom root cap cells. So here's a root cap cell. We're showing you gravity going towards the bottom of the screen. And so amyloplasts are pulled to the bottom of the cells by gravity. So if you were to turn the cell sideways, then they would all kind of line up at the bottom over here. And then if you turned it upside down, then all the cells would end up here because the cell's been turned upside down and now they'd be facing the floor. Okay. So the position of the amyoplast activates sensory proteins located in the plasma membrane, which initiates the gravitropic response, right? And by that, I mean, if you're talking about a root or a shoot, that means it's going to change the position. So it's going to grow into a different, or move in a different direction. So to give you an example of this, so here's a root. So imagine the root's in its normal position the way it should be, growing down, with gravity. So it's growing with gravity. So what happens is auxin is also produced in the roots. Remember, it's got those starch, those starch granules too that also detect gravity. And so normally what's happening is auxin gets produced in the root tip. It gets distributed throughout the root, but mainly, it's continuing to do its growth straight down with gravity. So this is the normal distribution. But if you were to take that root and turn it sideways, like you see here, so auxin initially is produced in the center of the root and then gets distributed throughout the tissues. But now a combination, so the action of those starch granules in the root cap, they're going to get redistributed, sensing that gravity is happening over here, right? Not over here. And so what's going to result is that the auxin is going to get redistributed in the root tip. So instead of being sort of evenly distributed, it's going to be distributed onto this side. And unlike what you see in shoots, this is a really important point. So in shoots, auxin initiates cell elongation. Okay, it causes you to bend towards the sun. In this case, what happens? Auxin is actively redistributed. And by the way, don't misunderstand this term. It doesn't actually use ATP. It just means that it gets redistributed, not using ATP in the root. And what it's going to do is it's going to inhibit growth. So what happens is auxin is going to end up on this side that's facing in the direction of gravity, but it's these cells that are lacking auxin that are then going to bend, and that's what's going to cause the root to then begin to bend down to grow towards the gravitational pull. So whereas before, auxin is what made the shoot, right? Auxin is what made the shoot bend towards the sun. In this case, it's the lack of auxin that makes it bend towards gravity. So that means auxin has a differential effect on root tissue compared to what it does in shoot tissue, which is a very important point. We'll talk about that more on Tuesday. Okay, I think this is my last objective. So objective 5. So we're going to talk about how plants respond to wind and touch. Uh-oh, you're not going to be able to hear the sound, I don't think. Hang on. How am I going to do this? Let's see if this works. I'm going to see if I can turn off my headphones. Oop. Okay, and let's see if you guys can hear this. I'll turn the sound up on my computer.
I'm a neuroscientist, and I'm the co-founder of Backyard Brains, and our mission is to train the next generation of neuroscientists by taking graduate-level neuroscience research equipment and making it available for kids in middle schools and high schools. And so when we go into the classroom, one way to get them thinking about the brain, which is very complex, is to ask them a very simple question about neuroscience, and that is, what has a brain? When we ask that, students will instantly tell you that their cat or dog has a brain, and most will say that a mouse or even a small insect has a brain, but almost nobody says that a plant or a tree or a shrub has a brain. And so when you push, because this could actually help describe a little bit how the brain actually functions, so you push and say, well, what is it that makes living things have brains versus not? And often they'll come back with the classification that things that move tend to have brains. And that's absolutely correct. I mean, our nervous system evolved because it is electrical, it's fast. So we can quickly respond to stimuli in the world and move if we need to. But you can go back and push back on the student. You say, well, you say that plants don't have brains, but plants do move. Anyone who's grown a plant has noticed that the plant will move and face the sun. But they'll say, but that's a slow movement. That doesn't count. That could be a chemical process. But what about fast-moving plants? Now, in 1760, Arthur Dobbs, the royal governor of North Carolina, made a pretty fascinating discovery. In the swamps behind his house, he found a plant that would spring shut every time a bug would fall in between it. And he called this plant the fly trap. And within a decade, it made its way over to Europe, where eventually the great Charles Darwin got to study this plant, and this plant absolutely blew him away. He called it the most wonderful plant in the world. This is a plant that was an evolutionary wonder. This is a plant that moves quickly, which is rare, and it's a plant that's carnivorous, which is also rare. And it's in the same plant, but I'm here today to tell you that's not even the coolest thing about this plant. The coolest thing is that the plant can count. So in order to show that, we have to get some vocabulary out of the way. So I'm going to do what we do in the classroom with students. We're going to do an experiment on electrophysiology, which is the recording of the body's electrical signals, either coming from neurons or from muscles. And I'm put some electrodes here on my wrists, and as I hook them up, we're going to be able to see a signal on the screen here. And this signal may be familiar to you. It's called the EKG, or the electrocardiogram, and this is coming from neurons in my heart that are firing what's called action potentials. Potential meaning voltage, and action meaning it moves quickly up and down, which causes my heart to fire, which then causes the signal that you see here. And so I want you to remember the shape of what we'll be looking at right here, because this is going to be important. So this is a way that the brain encodes information in the form of an action potential. So now let's turn to some plants. So I'm going to first introduce you to the mimosa, not the drink, but the Mimosa pudica. And this is a plant that's found in Central America and South America, and it has behaviors. And the first behavior I'm going to show you is if I touch the leaves here, you get to see that the leaves tend to curl up. And then the second behavior is if I tap the leaf, the entire branch seems to fall down. So why does it do that? It's not really known to science. One of the reasons why could be that it scares away insects or it looks less appealing to herbivores. But how does it do that? Now that's interesting. We can do an experiment to find out. So what we're going to do now, just like I recorded the electrical potential from my body, we're going to record the electrical potential from this plant right here, this mimosa. And so what we're going to do is I've got a wire wrapped around the stem, and I've got the ground electrode where? In the ground. It's an electrical engineering joke. All right. So all right. So I'm going to go ahead and tap the leaf here, and I want you to look at the electrical recording that we're going to see inside the plant. Whoa, it's so big. I got to scale it down. All right. So what is that? That is an action potential that's happening inside the plant. Why was it happening? Because it wanted to move, right? And so when I hit the touch receptors, it sent a voltage all the way down to the end of the stem, which caused it to move. And now in our arms, we would move our muscles, but in the plant doesn't have muscles. What it has are water inside the cells, and when the voltage hits it, it opens up, releases the water, changes the shape of the cells, and the leaf falls. Okay, so here we see an action potential encoding information to move, all right? But can it do more? So let's going to find out. We're going to go to our good friend, the Venus flytrap here, and we're going to take a look at what happens inside the leaf when a fly lands on here. So I'm going to pretend to be a fly right now, and here's my Venus flytrap. And inside the leaf, you're going to notice that there are 3 little hairs here, and those are trigger hairs. And so when a fly lands, I'm going to touch one of the hairs right now. Ready? 1, 2, 3. What do we get? We get a beautiful action potential. However, the fly trap doesn't close. And to understand why that is, we need to know a little bit more about the behavior of the fly trap. Number one is that it takes a long time to open the traps back up, about 24 to 48 hours if there's no fly inside of it. And so it takes a lot of energy. And number two, it doesn't need to eat that many flies throughout the year. It only needs to eat a handful. It gets most of its energy from the sun. It's just trying to replace some nutrients in the ground with the flies. And the third thing is it only opens, then closes the traps a handful of times until that trap dies. So therefore, it wants to make really darn sure that there's a meal inside of it before the fly trap snaps shut. So how does it do that? It counts the number of seconds between successive touching of those hairs. And so the idea is that there's a high probability if there's a fly inside of there that they're going to be clicked together. And so when it gets the first action potential, it starts counting 1, 2, and if it gets to 20 and it doesn't fire again, then it's not going to close. But if it does it within there, then the fly trap will close. So we're going to go back now. I'm going to touch the Venus flytrap again. I've been talking for more than 20 seconds. So we can see what happens when I touch the hair a second time. So what do we get? We get a second action potential, but again, the leaf doesn't close. So now if I go back in there, and then if I'm a fly moving around, I'm going to be touching the leaf a few times. So I'm going to go and brush it a few times. And immediately, the fly trap closes. So here we're seeing the fly trap actually doing a computation. It's determining if there's a fly inside the trap, and then it closes. So let's go back to our original question. Do plants have brains? Well, the answer is no. There's no brains in here. There's not no axons, no neurons. It doesn't get depressed. It doesn't want to know what the Tiger score is. It doesn't have self-actualization problems. But what it does have is something that's very similar to us, which is the ability to communicate using electricity. It just uses slightly different ions than we do, but it's actually doing the same thing. So just to show you the ubiquitous nature of these action potentials, we saw it in the Venus flytrap. We've seen an action potential in the mimosa. We've even seen an action potential in a human. Now, this is the euro of the brain. It's the way that all information is passed. And so what we can do is we can use those action potentials to pass information between species of plants. And so this is our interspecies plant-to-plant communicator. And what we've done is we've created a brand new experiment where we're going to record the action potential from a Venus flytrap, and we're going to send it into the sensitive mimosa. So I want you to recall what happens when we touch the leaves of the mimosa. It has touch receptors that are sending that information back down in the form of an action potential. And so what would happen if we took the action potential from the Venus flytrap and sent it into all the stems of the mimosa? We should be able to create the behavior of the mimosas without actually touching it ourselves. And so if you allow me, I'm going to go ahead and trigger this mimosa right now by touching on the hairs of the Venus flytrap. So we're going to send information about touch from one plant to another. So there you see it. So So I hope you learned a little bit something about plants today. And not only that, you learned that plants can be used to help teach neuroscience and bring along the neuro revolution. Thank you.
That is so bitchin'. Okay. Okay, I think I got my headphones back on. Hopefully you can still hear me. Okay. Oh, dang it. There we go. So how amazing is that? So plants are so bitchin'. So what you were just looking at there was thigmotropism. So a plant's movement in response to touch, as you can see, was pretty fast. It's rapid, non-directional. It occurs when a touch receptor cell transduces a mechanical signal to an electrical signal. And so you just saw a great video that expressed all that. All I'm really saying on this slide is what that guy just told you about. So basically, proton pumps in the plasma membrane of a plant cell to create a membrane potential. And traveling from a sensory to a response cell, the electrical signals are propagated in an action potential. And so we've already talked about how this happens in animals, and it's a super quick response. But in plants, it takes quite a bit longer, about 2.5 seconds. Now, as he mentioned, there are, of course, no synapses in plants. So instead, those action potentials are going to travel through those plasmodesmata going from cell to cell. So hopefully you remember all about the plasmodesmata. So it's going to travel through those going from cell to cell until it gets to the target where it's wanting to go. And he actually just told you about this, but let me tell you a little bit more. So when the action potential reaches the response cell, the cells change shape by either swelling or wilting and moving the plant. And so what you're seeing here is sort of a schematic of cells, in this case right there at the intersection of the stem or a branch with the stem. And you can see the water that's in the cells. Now, normally it's not all congregated like that. It's just trying to indicate to you that there's water in the cell. And then when those action potentials hit the cell, what will happen is it releases the water from those cells, causing the cells to shrink. And in this case, it's causing the movement of the branch to sort of wilt downwards. So that's kind of what he was talking about there in terms of how they managed to move so quickly. And there's some other things too. So it turns out if you do experiments and you have plants that you touch a lot versus plants you don't touch at all, so it actually causes them to respond by modifying the transcription of certain genes. And then as a result, it produces stout, really stiff stems. So they're shorter, but they're better able to stand up more properly, whereas these guys, if kind of grown unchecked and not touched, they get kind of wilty, flexible, less stout stems. So that type of response is called thigmomorphogenesis. And again, the result is the production of new proteins that stiffen cell walls to produce short, stocky plants. There's probably, that's one aspect of probably what's happening with those people that, like, you trim bonsai plants or other kind of plants and shape them. There's an aspect of thermomorphogenesis to that. It's not the entire explanation for how you get those things. That's a much longer story that we're not going into now. Okay, that's all I got for you. I'm going to shut this off now, and we'll talk more about this, and then we'll move on into defense on Tuesday. And I'll try to make some video lectures of that material too. I just don't know if I'm going to be able to push them out quickly today, but I'll try.